So what I would like to do is to start with an introduction to MEG and EEG analysis, MEG and EEG data, the analysis of this data, and the FieldTrip toolbox. So what is FieldTrip? FieldTrip is a MATLAB toolbox for the analysis of time series data, and we've developed it for EEG and for MEG, but we also started using it for animal electrophysiology, and nowadays we're also starting using it for NIRS, sometimes for fMRI. Basically any type of time series data can be analyzed using FieldTrip, but there's a strong focus on EEG and MEG because of the special properties that they have. FieldTrip can analyze data from many different file formats, from many different acquisition systems, and the toolbox contains algorithms for spectral analysis, for source reconstruction, statistics and connectivity, and all sorts of things that you need around it. The outline of the talk is that I want to start with explaining what kind of signals we are recording from the brain. And then I will explain what the temporal and spatial features of these signals are, because that's what we typically focus on in the analysis. I will then explain how we can analyze these signals using the FieldTrip toolbox, and at the end I will give a little bit of background on the FieldTrip project. But let's start with the signals that we're looking at. So what produces the electric currents and the magnetic fields? So we know that we have neurons in the brain, and if we look at the presynaptic neuron, it arrives at the dendritic tree, like the axon, and arrives at the synapse at a dendritic tree of a postsynaptic neuron. And what happens is if you have a presynaptic action potential, signal is transmitted to the postsynaptic neuron, and that causes a postsynaptic potential. And if you look at detail, the presynaptic action potential will release neurotransmitters, and these neurotransmitters will cause small currents to flow over the cell membrane of the postsynaptic neuron. And these small currents, they cause a postsynaptic potential. This should all be familiar to you. Okay, so there's different types of potentials that can occur in a postsynaptic neuron. First of all, if the release of neurotransmitter is large enough, then that might trigger an action potential. But action potentials are very short-lived. They're typically only a few milliseconds, and they have a biphasic pattern. So if we look at the action potentials from many neurons, it is very unlikely that they will be aligned exactly in time. So we might have one neuron receiving an action potential, producing action potential here, and a neighboring neuron producing action potential a little bit later. So it means that with action potentials, we actually don't really expect to see them at the scalp level, because they tend to cancel out, because they are poorly synchronized. There is some indication that we can see broadband activity, and the broadband activity that we're observing with MEG and EEG might actually relate to action potentials. But at this moment, the main notion is that we're mainly sensitive with EEG and MEG in picking up excitatory or inhibitory postsynaptic potentials. So basically the activity at the synapse is not large enough to elicit an action potential in the postsynaptic neuron, but a little bit of current will flow, so the postsynaptic neuron will depolarize or will hyperpolarize a little bit. This causes some current to flow, and that current is relatively long-lived. And this relatively long-lived current allows for easy summation, spatial summation of the other neurons in the vicinity of the neuron that we're looking at. So if we now look at the brain, and if we look at a very small section of the brain, let's say this part, then this is, with a specific cell staining, the type of structure that we see. So what we see is we have different layers in the brain, different layers in the cortical sheet, and in layer 4 we have the layer 4 and layer 5, that's where we have the cell bodies of the pyramidal cells. And these pyramidal cells, they have very long dendritic trees that extend towards the surface, and these pyramidal cells, they have a very nice topological arrangement, so they're nicely aligned, which means that if multiple pyramidal cells receives input, currents start flowing in the same direction. So here you can see these pyramidal cells. What happens is that we will have electric currents flowing along the dendritic tree of the pyramidal cells, and that produces EEG in the form of these electric currents and MEG in the form of the magnetic fields. Of Of course, this is a very local phenomena. So what you should think of is, well, you have a small pool of neurons. The small pool of neurons is creating a small current is created in this pool of neurons, and what will happen is that this current will passively flow through the rest of the tissue, because the brain as a whole is electrically conductive. So that means that ions will just flow through the tissue, will flow through white matter, but also through the skull, through the skin. And if we apply electrodes to the skin, we can record these currents as potential differences between these electrodes. So on the one hand, we have propagation of action potentials and propagation of postsynaptic potentials, which is an active process that involves the neurons. On the other hand, we have the propagation of the currents. And the propagation of the currents is just a passive process. It's the same process that we tap into if we do transcranial direct current stimulation. We just have current flowing through the tissue. And that's the current that we're recording with the EEG. If we consider Maxwell equations, then we know that with each current there's a magnetic field associated. And very important is that if we have very important for you to remember is the right hand rule. So if we have a current that is flowing in a certain direction, then with the right hand rule you can see which direction the magnetic field is flowing. And that's really useful. It's something that really helps you to understand the spatial topology. So like on this side the field is going out, but on the other side the field is going in. So it's curving around the current. And that means that if we have a small amount of current that is generated by a small pool of neurons, that will produce a magnetic field which can be picked up outside the brain. Tomorrow in the forward and inverse modeling lecture, I will explain that it's not only the primary current, not only the currents inside the neurons, but it's also the secondary currents that are flowing through the volume that contribute to the magnetic fields. The technical challenge of MEG is that we need very sensitive magnetic field detectors in order to record these magnetic fields. For that we're using superconductive sensors. And furthermore, the fields from the brain are very small, which means that even if we're able to pick them up, they're swamped in environmental noise. So that's why we need shielding, and we need a very smart sensor design. And that's what I now want to explain a little bit further. If you look at an MEG sensor, and let me take the simplest MEG sensor that we have, that's the magnetometer. The magnetometer is basically is a small loop, and all the magnetic field that passes through this loop is being sensed. The way that the magnetometer works is that we immerse it in liquid helium. So like we make a small niobium loop, we immerse it in liquid helium, and if you cool down niobium to a temperature that's close to 0, it will become superconductive. And if such a loop is superconductive, it basically means that there's no current, there's no resistivity. So the field that is coming in here will immediately be pushed out again, because a current will be generated and that current has no resistivity. But the thing is that here the field can be coupled in, and over here the field can be coupled out. So it basically means that we have one loop that senses the magnetic field, and we have another small loop to which we can attach our squid. And the squid is a superconductive quantum interference device. It's also a loop, but the squid has two Josephson junctions, like it has two weak links, and over these weak links we get a superconductive effect, the Josephson effect, which causes the current or the potential over this loop to fluctuate depending on the amount of magnetic field that is passing through the loop. So with an MEG system we're not using the squids to directly measure the magnetic field, but we're always coupling the squids to a magnetometer. It's the magnetometer that allows us to pick up activity from a much larger area and thereby amplify the activity. The squid has a very small surface area, the magnetometer has a much larger surface area. Since we have, so let me first explain the shielding. So we have these squids. They're sensitive. Squids coupled to magnetometers. These are very sensitive to the magnetic fields from the brain, but also to environmental noise. So that means that we have to put them in an environmentally shielded room. This is the first magnetically shielded room, which was built at the end of the 60s at the MIT, where the first MEG measurements were done. And what you can see is that it's more or less a sphere, because a spherical shielded room is most efficient in terms of shielding. Nowadays what we have is we have square rooms, because we can afford much thicker shielding. And this is a view of the magnetically shielded room that was built at the NACMEG in Stockholm at the Karolinska Institute 2 years ago. And this is actually very similar to the MEG rooms that to the shielded rooms that you will have on your side, or the one that we have down here, which we'll see after the coffee break. The difference is that here you can actually see how the room is made, because there's no cover on the walls yet. So what you see is that there's thick plating, and this plating is from new metal, and new metal has a property that magnetic field is absorbed by it. So what we have is a plating of new metal combined with plating of aluminium. The new metal is very efficient at absorbing low frequency magnetic fields. The aluminium is very efficient at attenuating high frequency magnetic fields. And then typically we will have 3 layers, like 2 layers of new metal with 1 layer of aluminium in between, but that varies a little bit. Besides the passive shielding that the new metal of the magnetically shielded room provides, we can also use an active shielding. So what you can imagine is using Helmholtz coils, and use the MEG system to record not only brain activity but also environmental noise, and then using these Helmholtz coils to do real-time feedback of the environmental signal. And that's something that we're not using here in Nijmegen with the CTF system, but that is something that's being used by the Elekta MEG systems. You can think of this as a way of reducing the fields. That's something that can be done inside the magnetic shielded room, but it can also be done outside the magnetically shielded room. So if you're working with an Elekta system, you should be aware that there's like 2 types of active shielding are being used. One that has the shielding outside the room, and one that has the shielding inside the room. The one that is outside the room basically tries to reduce the environmental, the fluctuation in environmental magnetic fields. So for example, it tries to compensate for the Earth magnetic fields and for the vibrations that the room or the building might have. The internal active shielding will also record activity from the brain itself, which means that these internal active shielding coils, they will try to attenuate not only environmental noise but also brain signal. That's of course not a desirable property, so that's something that you always have to compensate for in software. So if with an Elekta system you do a recording with internal active shielding, you always have to use max filter in order to remove the compensation of the brain data. That's just like something that you have to remember. Besides using shielding of the room to try to suppress environmental noise, we can also exploit the sensor design. So what I already showed you is that we have magnetometers as the simplest sensor. A magnetometer is basically a loop, but typically it's a few centimeters large. And with the magnetometer what we have is we pick up the magnetic field that is flowing through the loop. If I assume that I have a dipole pointing out of the screen, so it's right hand rule, so it's pointing out of the screen, so that means that current is flowing like this. If I now position this magnetometer over here, I will have a flux which is going in to the top. If I now move the magnetometer a bit, what you can imagine is that there's a little bit of flux going out, but also a little bit of flux going in. So that means that the total flux that is being picked up by the magnetometer is 0. And if I move it further along this line, you can imagine that the flux is going to point downward. So that means that with this magnetometer, depending on where I put it, I'm going to see a positive no magnetic field or a negative magnetic field. So that is a general feature. That's something that you probably are aware of. We see dipolar patterns. This is the reason why we see dipolar patterns. But it's also a feature that we can exploit by using gradiometers. So what I'm showing here is a planar gradiometer, and it consists of 2 loops. And what you should notice is that the direction of the loops is different. So this loop is going in this direction, whereas this loop is going the other direction. So that means that these 2 loops, they have a sensitivity in different directions. So at this moment there's a small amount of positive going flux in this direction, but there would also be a small amount of positive flux going in here. So that means that netto, this one being weighted positively, this one being weighted negatively, the amount of signal that would be picked up is a bit smaller than only the signal that is picked up by this loop. However, if I consider environmental noise, the environmental noise will have a spatial distribution which is much coarser. So typically it will be the same at both loops. So the environmental noise will have, in this example, downward going field here, but also downward going field here, which means that environmental noise will be cancelled out. If we consider the sensitivity profile of the planar gradiometer, if we shift it to sit directly on top of the source that we're interested in, what you see is that we will have a downward going field here and an upward going field here. There's a third type of sensor, and that's the axial gradiometer. And with the axial gradiometer, it's also measuring the gradient, but it's measuring it along the axis of the field, whereas this is recording it in the plane of the coils. So with the axial gradiometer, we have the same characteristics. So this one is picking up a large field, this one is picking up a smaller field. So the net effect will be that a little bit of this will be subtracted from this one. But again, if we have environmental noise, that environmental noise is going to be efficiently cancelled. So that means that with the different types of sensors that we have in MEG systems, we have different sensitivity profiles. We have magnetometers. Magnetometers are used in the system for 4D neuroimaging, which is formerly known as BTI. They're not manufacturing them anymore, but there's still quite a few of them around. So you might have a BTI system at home, and then this is the type of sensor that it has, and this is the sensitivity profile. So it has a positive sensitivity at the flanks, or is positive or negative sensitivity at the flanks of the source, but it's a zero directly on top of the source. The advantage of magnetometers is that it's sensitive to sources that are further away. The disadvantage is that it's sensitive to sources further away. It's like magnetometers allow you to record activity from the deeper brain regions, but magnetometers are also sensitive to environmental noise. So that's why most of the sites that have an MEG system use either axial gradiometers or planar gradiometers. Axial gradiometers have a sensitivity profile that is slightly sharper, but it also is zero on top of the source that you're interested in. But what you see is that if you move away from the source of interest, the signal is attenuated, which also means that if you have a source of no interest that is far away, it will be attenuated. It's considered environmental noise and it will be suppressed. If you consider a planar gradiometer, it's also attenuating signals from far away, but it has its sensitivity which is maximum on top of the source of interest. And if you move it slightly away, it will have a negative sensitivity profile. So why is this important? It's important because these are really the signals that you're going to work with. These are the signals that are stored on disk by the MEG system that you have. And you might have a different MEG system than the one that we have here in Nijmegen. So that's why for you it's important to understand and to appreciate the characteristics of the sensors, of your sensors. It also has some effect on how you want to process the data. For these ones and these ones, they can typically be processed in a very similar manner. They have a very similar sensitivity profile. But with planar gradiometers, you just have a very different sensitivity profile, so the interpretation of spatial figures will, for example, will be very different. And that is something that we're going to explain a little bit further in the hands-on session this afternoon. Okay, let me move on with the temporal and spatial features of the signals that we're considering. Of course, we've identified a lot of you are working with cognitive research questions where you would consider an event, like a cognitive event or a stimulus. And what happens is that if you stimulate the brain, you will get cortical activity. So if you just consider the brain is not doing anything, you present an event, and you get a response. And that is something that we can study in the presence of noise by repeating the same event, the same stimulus, many, many times. So if we repeat the same event over many trials, and if we assume that we have the same cortical response in each trial, then what we can do is we can average the data that we observe in each of these trials to give us the averaged event-related field. And the nice thing about this averaging procedure is that it will remove the noise. And actually here I've shown a moderate amount of noise. You can still see the average, the event-related response in each of the individual trials, which is uncommon. Like normally you would not see this. Like in real situation you won't see it. But the consequence of the averaging is that anything that is not synchronous over all the trials will be removed, and anything that is synchronous over the trials will stay. Another type of response, however, that we can see if we present an event is one in which the activity is time locked to the event, but where it's not phase locked. So in some of the trials it would start by going up, whereas in some other trials it would start by going down. And that means that if we consider this type of activity, and if we use the same trick of averaging over trials, the consequence is that we're not going to see any cortical response. To get this type of activity visible in our data, we have to use time frequency methods, which is going to be explained this afternoon. So if we consider the signal characteristics and how they translate into analysis methods, we know that we have the time course of the activity. For that we often use event-related potentials or event-related fields. We can look at spectral characteristics, so we can look at the power spectrum, or if we are interested in spectral characteristics around an event, we would look at the temporal changes in power, and we would be looking at time frequency responses. So this is all irrespective of the location. So this is the type of analysis that you can perform for EEG, for MEG, but it's also the type of analysis that you can perform for intracranial EEG, like if you can insert an electrode in the brain, or for ECOG data. But quite often we're doing experiments in healthy human subjects, which means that we cannot directly go into the brain, but we have to record on the outside. And that means that we have the skull and the scalp in between, and we have quite some blurring due to the skull and the scalp. So what we also have to consider is the spatial distribution of the activity of the head. And for analyzing, for exploiting the spatial distribution, we use source reconstruction techniques. And that's the topic for tomorrow's lectures. So all of these methods are relying on the temporal aspects, and all of these methods are relying on the spatial aspects. And often they're not completely separate from each other. So like in analyzing the spatial aspect, we can still make use of the temporal aspects, and vice versa. But this is just something that you have to, in general, have to keep in mind. There's two aspects to the data, and sometimes you want to capitalize on one, sometimes you want to capitalize on another. So far I've been talking about a single source or a single type of activity. But actually what we have, especially in cognitive experiments, is that we will have activity in many brain regions at the same time. So if we consider activity over here, then that activity was going to be most pronounced over occipital electrodes. If we simultaneously also have a more frontal process, for example, which has a much higher frequency content, then that's going to be most pronounced on over frontal electrodes, frontal sensors. But what we have in general is that we will have an overlap of activity. So electrodes that are in between will see both sources. And here I'm exaggerating a little bit, because if you just consider what these electrodes are going to see, then depending on the choice of your reference electrode, but also depending on how deep the sources are, all channels are going to see the activity from all tissue in the brain. So this frontal channel also sees a little bit of this occipital activity, and this occipital channel also sees a little bit of this frontal activity. So that means that in order to analyze EEG and MEG data, you always have to be aware of that we have a superposition of source activity. And disentangling this superposition, that's basically the important challenge that you have to deal with data analysis. To separate activity of different sources, but also of noise, like we have physiological sources of activity, like the heart. The cardiologists in the hospital would consider that their signal, we consider it noise. We have muscle activity, the neurophysiology people would consider that a signal, we consider it noise. We have cars driving by, so there's a lot of different sources of noise. They all have their own spatial and temporal characteristics. So we are interested in a specific type of signals. So what we do is we use temporal aspects of the data at the channel level to try to suppress noise and to capitalize on signal. So we use event-related field latencies. We can look at difference waves. Sometimes we can filter the time series, or we can use spectral decomposition. In order to separate activity of different sources, we can also use the spatial aspects. So what we can do is we can make a volume conduction model of the head, and using such a volume conduction model, we can estimate the source models. we can do source model estimation, trying to find the time series of the source at the cortical level rather than at the channel level. Is this all clear? Okay, because then I want to move on and to explain you how we do these types of analysis using FieldTrip. So some FieldTrip basics. FieldTrip is a toolbox. FieldTrip does not have a graphical user interface. Like, it really comes as a whole set of tools, and you have to combine and mix and match these tools. FieldTrip tools are basically the MATLAB functions. So if you look at the MATLAB function, or if you look at the functions inside the FieldTrip toolbox, what you see is that they have each function has a name. It takes a number of arguments as input, and it produces an output argument. Some of the FieldTrip functions do not produce output, like not as a data structure, but they produce a figure. Some of the functions do not take input arguments, such as the pre-processing function, which reads data from disk and then starts doing something with it. But in general, what you see is that all of these define how the function should behave. And the strategy that we use in FieldTrip is that we try to make this configuration structure human readable. So we try to make it such that config channel is a list of channel names that you can recognize. So rather than using channel indices 1, 2, 3, 4, we use channel names. Using channel names allows you to remove a channel, a bad channel, without having to reconsider that all the channels have to be remapped. So that it facilitates bookkeeping. Also with other parameters such as frequency of interest limits, FOILIM, we try to use them as much as possible in human understandable characteristics. So this is frequency of interest expressed in hertz, from 1 till 70 hertz. So that means that with the configurations you have a human readable form of how the function is going to behave. And if you compare this to default MATLAB, then this is how FieldTrip behaves. So we have a function that takes a configuration, that takes an input data structure, produces output data, and the configuration has key value pairs. So we have a key, and the key has a certain value. And you might recognize that some of the more advanced MATLAB functions also have these key value pairs, but they often specify them like this. So we have a function with a number of fixed input argument, and then you have key value, key value, key value. So functionally it's equivalent. So we could have implemented it like this. The reason for implementing it like this is that we like to work with scripts. So we like to put all of these things that we're writing in terms of data analysis, we would like to put it after each other. If you do it like this, you get very long lines, which are very difficult to read. If you do it like this, you get all your lines nicely underneath each other, and you can very easily comment out one line, change one of the options. So that means that the script that you're producing is human readable. And that's for us very important, because it's the scripts that allow you to analyze data. I said that we're using functions, and we have a whole lot of these functions. And the way that you should think of these functions, that you put them together in an analysis pipeline. So you would start with FT pre-processing. And FT pre-processing reads MEG data from disk, it applies some filters, and it produces an output data structure. And once you've done FT pre-processing, you could do reject analysis, or you could, this is for example how you call it. So you specify the data set, you specify the filters, and then you call FT pre-processing. And then next you could do FT-FRAC analysis, where you would specify the method, multi-table method with fast Fourier transform, you would specify the frequency of interest limits, and then you would call FT-FRAC analysis. So by this way you're constantly calling all of these FieldTrip functions, and each of the functions has its own configuration. And it's important to keep in mind that there's not a single configuration that links all the FieldTrip functions together. It's your script that links all of these FieldTrip functions together. So the best practice also is in your script to always start with an empty configuration, because this configuration applies to this function and not to this function. Besides the configuration, FieldTrip works with data structures. And this is an example data structure for raw data. So what you see is that it has labels, channel labels, 151 channels. It has trials, 87 trials. It has time axis. Each trial has its own time axis, and it has some additional fields. If you then look at the result of time lock analysis, which computes the event-related potential or the event-related field, you see that it again has labels, but now instead of having trials, it has a single average. And here in this single average, you can recognize a matrix, 151 channels by 900 time points. That's basically the matrix that contains the average. And sometimes we also compute other stuff such as variance. So that's what you would also encounter here. And then here you see the time axis. Here, since we only have a single evaluated field, there's only a single time axis. So what you can imagine is that this time axis together with this labels describes this average. And that's what we often represent in the dim ord. So the dim ord is a field that facilitates bookkeeping, and it specifies the order of the dimensions in the data. So here the dim ord is Chan by time, which means that Chan, we have 151 channels, time we have 900 time points. So any matrix that is present in this data structure, which is 151 by 900, will be interpreted as a channels by time matrix. For this one, it's not so difficult to see how the data is organized. But once you start working with multi-tapered spectral analysis that are time varying, you end up with 4-dimensional, sometimes even 5-dimensional data structures. And then keeping track of what is represented in which dimension is quite important. What you also see is that each of these data structures has a configuration. Let me explain that in a little bit more detail. So the input configuration of a function specifies the parameters of the algorithm that's contained within that function. The output config that you see in the data structure keeps the history. So if you look at this function call, then we have data out, which depends on data in 1, data in 2, etc. And here we've specified how this function should behave. And data out is of course the data structure with the numeric results, but it also contains data out.cfg. And data out.cfg is basically this configuration, which contains your settings, but also the defaults. So any defaults that we used in this function will also be in there. So that means that after having done the computation, you can look at exactly what the computation, what the default settings for the computation were. Of course, these ones have also have their own history. So that means that data out.cfg can contain information from the previous function. So data out.cfg.previous1 is the configuration that was contained in here, etc. So the details of the computation are kept with the data, but the previous data is not kept. Like FieldTrip is not managing data for you. FieldTrip is only managing history. So it is up to you to organize these data structures. Like you can decide whether you want to save them on disk or keep them in memory. You can also decide how you're going to call them, like how you're going to distinguish your experimental conditions. But what FieldTrip will do for you is that it will keep track of the history using config.previous.previous.previous. And that's a very useful feature because it means that after having done a lengthy analysis, you might have lost track of what you've exactly done in a previous step. So we have a function which is called FT analysis pipeline, and FT analysis pipeline will show you a graphical representation of all the steps that you've taken. And that's important because if you write your paper, you want to be sure that you've done your analysis according to the way that you describe it in the method section. Okay, so how do we use this in script? Well, we would start, for example, with FT preprocessing. So we would write a small section that pertains to preprocessing. Then we would do FT frec analysis, write a small section, and then we would FT frec statistics, and then we would continue with plotting, or we would like branch off to a completely different type of analysis. And it's these scripts that you manage yourself, and it's these scripts that you store on disk, and that's where you basically put the structure of your analysis. And what we quite often have is analysis over multiple subjects. So that allows you to use the scripting facilities of MATLAB to first write your analysis for a single subject, and then you just add a for loop for each of your subjects, for each of your conditions, do the same computations. And this is nice because it means that you can first write your script for a single subject, and then you can just add for loops, and then you switch it on in the evening, and it will be churning through all the data during the night. And in the morning, you're probably going to find that it ran out of memory. Because what I'm doing here, I'm storing everything in like local MATLAB variables. So actually what we do most often is that in such a script, we write the data to a file. So that means that in the next morning you will find all these files on your disk. Of course, if you script your analysis like this, It also means that you can very easily use distributed computing. And here at the Donders, at the DCCN, we have a compute cluster, and on this compute cluster we have the Torque Q-sub system, and FieldTrip includes a small toolbox, the Q-sub cell-phone toolbox. With Q-sub cell-phone, you can very easily do distributed computing. So what you see here is that here, for each of the subjects, for each of the conditions, I'm preparing a configuration. And this configuration basically specifies what the function should be doing. So this is the configuration for frag analysis, and this is the configuration for FT preprocessing. And what I then can do is I can do Q-sub cell-phone, FT preprocessing on this configuration. So this is a cell array, number of subjects times number of conditions. And what this will do is it will call FT preprocessing in parallel on each of these individual things, which means that you don't even have to wait the night, because it will just run as fast as the analysis on a single subject. And then once you've done the preprocessing on all your subjects, you would call FT frag analysis, which would call frag analysis on all the subjects. If you do this type of distributed processing, it is important to really consider how you save the data on disk, because this really blows up data very quickly. So what you see here is that I'm using the option config output file, which specifies a FieldTrip function, like any FieldTrip function, that it should write its result to disk. And in the configuration for frag analysis, I'm specifying config input file, and here I'm saying, well, the input file is the same as this output file. So what I'm specifying is like write all the preprocessing results to disk in this naming structure, and then read from all those files. So this is using Q-sub cell-phone. This is a FieldTrip toolbox for distributed computing. Like it's not only used inside the Donders, but also some various other sites. But if you have the MATLAB distributed computing toolbox, you could also use df-eval, and df-eval works exactly the same. So that means that distributed computing should be really easy if you structure your scripts accordingly. Everything clear so far? Okay, so FieldTrip is a toolbox. It has tools, and you combine these tools in building something. It also means that the data and the separate functions, the function that you're going to call in this data, are in your hands. So you write your script, and you decide how your script is going to be organized. And the scripts that you're going to write, they depend on properties from the data. Like MEG datasets tend to be much larger, so for MEG it's much more important to store all the intermediate data on disk. EEG often has fewer channels, lower sampling rate. So with EEG it can be much more convenient to read all the data from all your subjects into memory and to toss around that data in memory. It also depends on your programming skills, on your programming style. So scripts can be quite different between researchers. But the important thing is that the script corresponds to analysis protocol. That means that you can show your script to your colleagues. And you can share your scripts with the colleagues. And that's what we do a lot here at the Donders, where scripts are being handed over from one generation of PhD students to the next generation. And of course, scripts are not always so clean, and so you might not be able to reuse everything, but they do contain a lot of information about how people have worked in the past. And it's also something that you can directly discuss with your supervisor and with your colleagues. So besides only showing figures that contain the output of your analysis, which are often discussed with your supervisor, you can actually just take your scripts with you, or just have like a shared desktop where you can basically both sit on, and then you can look at the scripts, and then you can say, well, this script produces this output, and then you can start asking questions like, what would change in the output if I were to change this parameter? So FieldTrip is not so. FieldTrip does not have a graphical user interface, so you cannot very easily click around and try out different parameters. But FieldTrip allows you to try different parameters by changing the script, and that's also why it's important that FieldTrip keeps its own history. Like the data structure contains their own history, so even if you've changed your script like 10 times in a row, the data structures will still contain in config.previous exactly how they were computed. The FieldTrip toolbox has quite a lot of functions. I think it's more than 100 functions, which means that it can be a bit challenging to find the appropriate function for the type of analysis that you want to do. Of course, we have inside MATLAB, you can type help function name. All high-level FieldTrip functions start with the prefix ft underscore. That's something that we're going to use in the hands-on session. So like if you type help ft underscore, and then if you press tab, MATLAB is going to show you a list of all the FieldTrip functions. And we try to structure the functions in such a way that we, for example, all functions that have to do with frequency analysis, they start with frec. So if you do help ft underscore frec and then press tab, you're going to see a list of all the functions that pertain to frequency analysis. FieldTrip is open source. That also means that you can directly look into the function. So you can just type edit function name to see what the function is doing in detail. Of course, we have the FieldTrip website at fieldtriptoolbox.org. And on the website we both have reference documentation, we have tutorials. And we have the email discussion list. But I think something that's just difficult to point at is that there's often expertise in your local research environment. You know best what kind of data you're working with, and your colleagues know best what kind of data you're working with, because often you will be working as a team. That means that you should really also consider your local research group, the people in your room, and the people that are working on similar research questions, because you can approach them very easily, and you don't have to explain the whole story. Like, this is my paradigm, this is the type of data that I have, and I'm stuck on a stupid error in some FieldTrip script, because often the problems that you run into are very specific to a research question that you're working on. So that's why, if you consider asking for advice, this is probably where you should start. First ask your colleagues. If with your colleagues you can't figure it out, then you can search the website or ask on the email discussion list. Yeah? Okay, so let me also give a little bit of background on the FieldTrip toolbox. Who is the audience? Well, the audience is basically people like you, experimental neuroscientists, but we're not offering a graphical user interface. So we're targeting more dedicated and more ambitious researchers, like people that are willing to invest time in getting to know the tools. Once you know the tools, they're very powerful, but they're not so suited for master students or so, who only have a limited amount of time to quickly process some data. I think for certain groups of users, other tools are more appropriate, like tools with graphical user interfaces, where you can just basically click through your data and just very quickly get the reward of your analysis. But FieldTrip also targets the developers of other software packages. So we have been working together with the SPM team for quite some years now, also with EEG Lab. In a few weeks from now, I'll also be giving an EEG Lab workshop. EEG Lab is one of these toolboxes that has a that capitalizes on the graphical user interface. It really shows you the data at all steps of the analysis. And I think that makes it valuable for a certain type of users. We're also working together with companies such as BISA. I think BISA makes good products. A lot of companies make good products. If you have it in your lab, you can just exploit the product. If you don't have it, you don't have to. FieldTrip is also being used for brain-computer interfacing, and that's where we are working together with BCI 2000, which is a platform for online processing. FieldTrip's strength is not really in online processing. You can use FieldTrip to do rapid application development of online paradigms, but BCI 2000 is really much more capable of running robust online analysis. But we're also working together with teams that have a more specific research focus, such as Symbio and OpenMEG, which are teams working on volume conduction modeling without necessarily having a strong interest in all the signal processing aspects that we're also working on. And what we try to do with FieldTrip, or with the community where FieldTrip is one of the players in, is we try to make the code such that we can share. And of course what we're also targeting is developers of analysis methods. I'm one of these myself. So I'm using FieldTrip to communicate my new analysis methods to an audience of experimental neuroscientists. But at the same time, I'm also relying on the experimental neuroscientists to give me feedback on the methods that I'm developing. So there's a two-way interaction. It's not only that methods people are giving the experimental neuroscientists information, but it's also the experimental neuroscientists that give the methods people information. And it's really about bridging the gap between the different disciplines. I guess that a lot of you have already downloaded FieldTrip once, so have looked at it. And I want to explain a little bit more how the toolbox is organized. So if you take the end user perspective, then FieldTrip is basically, it's a toolbox. And it has FieldTrip main functions, those are the ones that always start with ft underscore and then something else. And they always have a configuration structure as the first input argument. And then FieldTrip has a lot of private functions. But if you start digging around in the FieldTrip directory a little bit more, you will see that these private functions are distributed over a whole bunch of like small mini toolboxes. So we have a small toolbox for file I/O. That's the one that we're sharing with EEG Lab and with SPM. We have a toolbox for forward modeling, for pre-processing, for multivariate analysis, for connectivity analysis, for distributed computing, and for a whole bunch of other things. And by organizing the toolbox in this way, we can more easily collaborate with other research teams. Sometimes it is also helpful for you to call one of these functions in a low level, but in general, you should start by calling the high level functions, because it's the high level functions that do the bookkeeping for you. So this is where we have functions such as FD pre-processing, frequency analysis, dipole fitting, source statistics, etc. And this is where we have functions such as FD read data. FD read data produces a matrix, and that matrix is basically channels by time. So FD read data does not do any bookkeeping for you. It's the FD pre-processing that does the bookkeeping. Sometimes it's more convenient to call the low-level function, but in general you should call the whole high-level functions. And we have FD compute lead field for the forward computation, or filtering functions, etc. So to summarize, I've explained you the type of signals that we're recording from the brain. I've explained the temporal and spatial features that these signals have, and how these features can be analyzed using the FieldTrip toolbox. And I've given you a little bit of background on the FieldTrip project, because FieldTrip is more than just the code, than just the toolbox. It's also the design of the toolbox, but also the user community that I hope you will all become part of. Yeah, and I think it will help if you understand the user community to get the best out of the analysis of your data. Okay, thank you.